Lab 5, Fetal Pig Dissection Part 3, Circulatory, Respiratory, and Nervous Systems. 3 experiments were performed. First, the difference in lime water cloudiness was tested under room air and exhaled air conditions. Next, the amounts of carbon dioxide in the air at rest and after exercise were compared with each other using phenolphthalein as a carbon dioxide indicator. Finally, the vital capacity for each person was recorded using a spirometer. The circulatory system transports nutrients, gases, wastes, hormones, and other materials between the systems in the body by capillary network. The total circulatory system is broken into 2 smaller systems, the systemic and pulmonary circulation pathways. The systemic pathways transport nutrients and waste, while the pulmonary pathway transports oxygen and carbon dioxide. The circulatory system between the fetal and adult animals differ because of anatomical and molecular differences. Placental fetuses receive all oxygen and nutrients as well as dispel waste through the mother. Because the fetus does not breathe, there is a hole between the heart atria, resulting in the mixing of oxygenated and deoxygenated blood. Also, the fetus lives in a low oxygen environment and has different hemoglobin than the adult. Fetal hemoglobin has a higher affinity for oxygen in order to retain the most oxygen from the maternal blood. It also prevents the release of oxygen until nearly deprived.
Oxygen is very necessary for sustaining the life of mammals. It is transported throughout the body by way of blood. The respiratory system, in humans and in other mammals, is responsible for delivering oxygen from the surroundings into the system for survival. It is composed of the lungs and the respiratory muscles that inhale oxygen through the oral cavity and down the trachea, and is responsible for exchanging and releasing carbon dioxide from the system when exhaling. The process of passive diffusion is used to exchange the oxygen captured from the surroundings with the carbon dioxide waste produced by the cells in the body. The alveoli are structures found in the lungs that enable this exchange. Under different conditions, we hypothesize that there will be a significantly different amount of carbon dioxide produced by the body and released in exchange with oxygen.
The nervous system is comprised of the brain, spinal cord, and nerves. The specialized cells that make up the system are called neurons that transmit signals throughout the body. It maintains homeostasis while monitoring and responding to external stimuli. It recognizes any changes in temperature, pH, hormone concentration, pressure, and other sensory inputs. The system responds to these changes by sending signals from the brain to the organ that converts the signals into action.
The methods for dissection from Lab Procedure by Buchholz in chapters 5, 6, and 8 from the second edition of the Dissection Manual for Fetal Pigs by Smith and Schenck were used to complete the dissection part of this laboratory. The parts of the pig that are important and were removed from this lab were the small intestines, large intestines, the heart, and the brain. The length of the intestines were measured and compared to the length of the pig. The ratio of length of the large intestine to pig length was 1.1, which is similar to that which was stated in Lab 5. The ratio of small intestine to pig length was 8.6 times as large.
The first experiment that was performed was comparing the room air to the exhaled air. Lime water, or concentrated calcium hydroxide, turns cloudy when it comes in contact with carbon dioxide. We predicted that our exhaled air will have more carbon dioxide than the room air. Eventually, the beaker with bubbled room air will turn cloudy, but only after a significant amount of time. Room air was bubbled through lime water for 1 minute using a pipette pump. Exhaled air was bubbled through the water using a straw. As expected, the lime water beaker with room air did not turn cloudy. After bubbling room air through the solution for another minute, no change was recorded. The lime water beaker with exhaled air did turn cloudy and opaque after 1 minute. Table 1 shows the observations for each condition. Exhaled air had a larger concentration of carbon dioxide than the room air. This indicated a difference in the amount of carbon dioxide under different conditions.
The effect of exercise on carbon dioxide production as compared to normal exhalation at rest was observed as well. The methods were followed as stated in Lab 5, but phenolphthalein was already prepared by the instructors. The hypothesis was that after exercise, the body would exhale more carbon dioxide. The data can be seen here in Table 2. As you can see, the exhaled air after exercise contains more carbon dioxide than the exhaled air at rest. Our hypothesis was correct.
The data from the carbon dioxide exercise experiment for the class was compiled in Table 3. The averages of each section's data for each of the 3 different conditions were used. The room air had significantly less carbon dioxide production. Comparatively, exhaled air after exercise produced more carbon dioxide than at rest. This was expected because the rate of respiration is faster after exercise and more carbon dioxide is produced by your cells. The high standard deviations in this case indicates a large range between the data values and there are more outliers present. The null hypothesis states that there is no difference in the amount of carbon dioxide in the air versus the amount exhaled from our lungs at rest or after exercise. The alternative hypothesis states that there is a difference in the carbon dioxide concentrations under different conditions. In order to analyze the class data in Table 3, an ANOVA, or Analysis of Variance, was performed in Excel. Figure 1 presents the ANOVA. The p-value produced is the chance that the data is produced by chance. The p-value for this data is 0.015869. If the p-value is less than 0.05, the null hypothesis can be rejected. However, if the p-value is greater than 0.05, the null hypothesis is failed to be rejected. In this case, the p-value is less than 0.05, so the null hypothesis is rejected. Therefore, there is a significant difference between each condition and the carbon dioxide produced. This is consistent with our hypothesis that after exercise, more carbon dioxide is produced than at rest, and room air has the least carbon dioxide out of the 3 conditions.
Vital capacity is the total exchangeable air in the lungs. As instructed in the lab procedure, the vital capacity of each group member was calculated with a spirometer 3 times. The averages were taken and the data lie in Table 4. These values were then compared to the standards for females at our specific heights. Our heights are 5'3" and 5'6", and the normal vital capacities for our heights is 3.1 and 3.4 respectively. However, my partner's calculated vital capacity was 3.5 and mine 3.2. We deviate from the normal. Table 5 shows the average vital capacities for each person in the lab. The average vital capacity for the lab members is 3.5 and the standard deviation is 0.247. There were both males and females performing this lab, and based on the chart for normal vital capacities for males and females, we do not think these values can be averaged because the normals for genders is different.
General conclusions can be drawn from these 3 experiments. It was demonstrated that there is a significant difference between the amount of carbon dioxide in the air and exhaled breath. Also, under different conditions, different amounts of carbon dioxide can be produced, and vital capacity depends on age, sex, and height, but often deviates from the normal.